I'm going to talk a little bit about some of the work that we've been doing on using microorganisms to promote health in a variety of ways. But to get you on my side, I'm just going to show a couple of slides. We haven't talked much about the nutritional aspects of cheese so far in this conference, but there are a number of studies that have shown that the cheese is actually good for you by providing a number of minerals and vitamins. This whole business about polyunsaturated fatty acids and CLA and the links with health. And there have been some studies that have shown that consumption of high-fat dairy products, including cheese, leads to a lower body mass index. So obviously I don't eat enough cheese, but it's also linked to, that's linked to stimulation of a number of hormones such as glucagon like peptide 1. There are also a number of other components of cheese that are good for you. The proteins and whey proteins are linked to immunostimulation and blood sugar regulation. And some of the branched-chain fatty acids contribute to muscle building. I'm going to talk a little bit about bioactive peptides because we've done a fair bit of work on them. But there's a whole bunch of health claims related to these bioactive peptides. But I'm going to start first by talking about some of our work on novel starter cultures. There are a group of polyunsaturated fatty acids called omega-3 fatty acids. And the two main ones are EPA, or eicosapentaenoic acid, and DHA, which is docosahexaenoic acid. So if you think some of the bacterial names are bad, then some of the fatty acid names are even worse. But these fatty acids are essential fatty acids. The human body doesn't produce them, so they have to be supplied by the diet. And especially DHA is essential for proper neural development in fetuses and young infants. And it's also been linked with a whole bunch of physiological problems such as cardiovascular disease. They're supposed to protect against cardiovascular disease. They have some anti-cancer properties. They help relieve arthritis and inflammation. They also have a role to play in alleviation of some of the symptoms of diabetes, and they've also been shown to be protective against HIV. So it's the old business of, I remember when I was young, my mother fed me cod liver oil, so that's why you eat cod liver oil, is to get your EPA and DHA. So the main source of these omega-3 fatty acids are fish oils. You're going to hold your hands up in horror. I'm going to talk about genetically modified organisms at an artisanal cheese factory. So, please, I think I'll hide behind. Anyway, what we've been doing is looking at some of the non-fish, the non-diet sources of DHA. And there are a number of bacteria that can produce both EPA and DHA. And what we've done is isolated the genes that are responsible for the production of the enzymes that make these fatty acids. And we've cloned these enzymes into both E. coli nisli and Lactococcus lactis. And Salmonella is the original organism from which we isolated these genes. So this is some of the profiles of our transgenic E. coli. So we have E. coli that can produce both EPA and DHA at a variety of temperatures, which is interesting because you may be able to use E. coli as a source of omega-3 fatty acids. But what is more interesting from our point of view is now we've been able to put those genes with over a large number of years and with a lot of effort, we've actually managed to put the genes that synthesize DHA and EPA into lactic acid bacteria. And the interesting thing is that these organisms, these lactic acid bacteria, produce DHA and EPA over a fairly wide range of temperature. So we've got the possibility of producing starter cultures and other useful organisms that are capable of forming these polyunsaturated fatty acids. So why are we doing this? Well, DHA forms about 8% of the brain by weight. It's obviously important for pregnant mothers. DHA is added to some infant milk formerly, and it's produced a lot from algae, and that market is worth about $1.5 billion US each year. We also have a product in Ontario called Dairy-O. This is a milk that is produced from cows that are fed fish meal to increase the polyunsaturated fatty acid levels in milk. So this is a sort of a natural way of producing an enriched milk containing polyunsaturated fatty acid. What we could maybe do with this technology is to produce cultures for producing silage that are rich in these polyunsaturated fatty acids, and then substitute this silage and other sort of forages for fish meal in the diet of cows. There's also the possibility of producing DHA probiotics, and that would essentially turn these polyunsaturated fatty acids from being an essential fatty acid in humans to non-essential fatty acids, if you can get colonization of the gut. So that's some of the bad research that we're doing to get that out of the way first. I mentioned we've been looking at bioactive molecules, and we've been doing a fair bit of work with probiotics. I'm looking at the bioactive peptides that are produced in two ways. One, as a direct product of the growth of these mainly lactic acid bacteria, and secondly, the peptides that are produced as a result of the breakdown of the proteins in milk, such as caseins and whey proteins. I'm not going to go into too much detail about this, but there have been a number of ways to try and explain how probiotics work. There's the whole bunch of agents that are produced by lactic acid bacteria that directly inhibit other microorganisms. There's the whole business about competitive exclusion. The idea being that probiotics line the gut and prevent some of the bad bugs sticking to the gut. There's also some evidence to suggest that they alter barrier functions of the gut and the immune system, and they can actually interact with the host epithelial cells as well and change some of the properties of the host cells. What we're interested in is number two, how if these bioactive molecules that are produced by probiotics actually can reprogram pathogens. And I'll explain what I mean by that in a minute. So we've asked questions, for instance, such as, can they prevent infection by E. coli 157? We've talked a little bit about E. coli 157, but it's a little bit more than 157. There are about 6 serotypes of E. coli that can cause hemorrhagic uremic syndrome and a number of other nasty illnesses. So it's not only 0157 that is problematic these days. It's other serotypes such as O26 and O111. what these two graphs show in effect is that these probiotic molecules switch off virulence genes in E. coli. We have a neat way of looking at expression of virulence genes in E. coli. We fuse these genes with what are called LUX genes. These are the genes that are responsible for bacteria producing luciferase, which is an enzyme that converts chemical energy into light energy. So if the genes are switched on, then the LUX genes are also switched on, so the organism lights up. So this basically shows we've added a number of sort of commercial probiotic cultures and to two different virulence systems in E. coli 157. Basically what we've shown is that there are a number of probiotics that switch off virulence genes in E. coli 157. RC14 doesn't work too well. But we've concentrated on Lactobacillus acidophilus LA5. And using a technique called RT-PCR, reverse transcription PCR, what we've done is to look at how some of the virulence genes in E. coli 157 are affected by these bioactive molecules. And basically what we've shown is that these bioactive molecules downregulate a whole bunch of genes, and I'll explain what those genes control by using an animation, if it'll work. This one. So this is a Howard Hughes animation. It explains how E. coli 157 colonizes the gut of humans. So what it does, it sort of settles on the epithelium. It sort of knocks down the microvilli, anchors itself with flagella, and then what it does is starts to synthesize some of the proteins, the ESPA, ESPC, and ESPD. And these proteins will actually produce a syringe. So these proteins form this structure which sort of punches a hole in the epithelial cell. And then you'll see a red darning cone-type molecule that comes down, and that's called the T protein, and that actually forms a receptor for the E. coli 157. So this T, or the translocating intamin receptor, actually the bacterium actually produces its own receptor molecule, and then that will sort of anchor the bacterium to the gut wall, and then there are enzymes in that are associated with these intamin receptors that then produce they polymerize actin. So you get a whole bunch of these little molecules are actin. So you get actin polymerization, and then this lifts the whole structure up to form these pedestal type structures that are typical of enterohemorrhagic E. coli infection. So having shown you that, what we've shown is that the bioactive peptides that we've isolated actually prevent the injection proteins, if you like, and the intamin receptor from being expressed. So it actually stops colonization of the gut with E. coli 157. it doesn't stop the growth of E. coli 157. It just stops E. coli from sticking to cells. And this is another way of showing that. This is some work with a tissue culture. So these are intestinal epithelial cells. This is the sort of natural cells without infection by E. coli 157. This is following colonization by E. coli 157. So those little dots you can see inside the cells are the actin polymers that you saw being formed. And this is what happens if we first give our probiotic bioactive peptides to the epithelial cells before we infect with E. coli 157. So you can see there's not much difference in the morphology between the healthy cells and the bioactive protected cells. I won't go into D. But what we think is happening is, we mentioned yesterday that bacteria actually talk to each other. What we think, we've got some good evidence to suggest that these bioactive molecules actually prevent E. coli 157 from talking to each other and producing the virulence factors that are necessary for colonization of the gut. So again, what this slide shows, it's fine to show it works in tissue culture and in vitro. It's nice to show that it works in vivo. We have a technique in the lab where we can actually image colonization of the gut of mice with E. coli 0157. Again, what we've done is used our luminescent strain of E. coli 157, and we have some imaging systems that allow us to detect light emission across the abdominal cavity of mice. So these are actually live mice. They've just been anesthetized and put in our imaging system, and those colored dots you can see on their bellies are areas within the gut where E. coli 157 has colonized. So this is after 3 days and 5 days of infection with E. coli. These poor little beasts didn't last 7 days. They all died before 7 days. The second row is what happens when we challenge with E. coli 157, and then we add our probiotic bioactives. So you can see that there's a fair bit of protection against colonization of the organism produced by giving these probiotics. So there's a therapeutic effect of these probiotic bioactives. And these mice were still alive after 7 days. And the bottom row is what happens when you feed the probiotic bioactives and then challenge with EHEC. And again, we get survival of the mice after 7 days and a reduction of colonization. So there's also a prophylactic effect of these peptides. And basically this is the same sort of experiment, but this time we've delivered the active fraction in yogurt, so that we show that we can protect mice by feeding them yogurt containing this bioactive peptide fraction. We haven't tried cheese yet. Mice don't like yogurt for some reason. They like cheese, or there's a perception that mice like cheese, but they don't like yogurt. But it works. So do these probiotic bioactives work with other organisms? Again, I'm not going to go into any detail, but in Salmonella, we've shown that it stops the expression of 2 genes that are responsible, again, for attachment of the organism to the gut and for regulating the the survival of Salmonella. And recently we've shown that this probiotic can protect against Salmonella infection in pigs. If you remember when I said that these bioactives don't kill or don't inhibit the growth of Salmonella, what they do is prevent Salmonella from sticking to the tissues of the large intestine. So you'll still see Salmonella inside the gut, in the contents, but you don't see any on the tissues, on the tissues of the large intestine. So it's indicative of the molecules preventing the organism from sticking to the gut. So that's the bioactives that are produced directly as a result of growth of the probiotics. We've also done some work on peptides that are formed during fermentation of milk. And again, this shows that these peptides have a role in stimulating the immune system. It shows the changes in production of what are called cytokines, and again, these are sort of signaling molecules that are used to switch on the immune system. Again, I'm not going to explain too much about these, but what these graphs show is that these peptides actually stimulate the immune system. And they also protect against infection in mice. So the line with the round bars are mice that we challenged with Salmonella. So, and these are the percentage surviving. So 7 days post-challenge, none of the mice that we infected survived in the control subset. But when we add our bioactive fraction that's produced in fermented milk, we get 100% survival after 7 days. So again, indicative of protecting the host against the ravages of Salmonella. These are the sequences of the peptides that we've isolated. Again, I don't expect you to take much away from this, but well, what's interesting is that we have this short peptide that's produced by degradation of whey, which has immunomodulatory properties. Usually it's peptides that are produced by cutting up caseins that are the bioactives, and this is the first time that a whey peptide has been shown to be involved in immunomodulation. So again, why are we doing this? Just for EHEC alone, in the US, there are about 73,000 EHEC cases each year with 60 deaths. We had one significant, that was actually a hard cheese that was produced in Alberta, which resulted in 11 cases of EHEC infections, and that drove that particular manufacturer out of business. We've had some unpasteurized milk cases as well. So for E. coli 157, the infection rate is about 0.7%, 0.7 per 100,000, and an economic cost per case is estimated to be about 3,000. On top of that, the European market for probiotics was projected to be about 10 billion euros in 2010. So by developing these probiotic bioactive peptides, we can maybe produce designer products with possibly some therapeutic uses, and we may be actually able to prevent shedding of E. coli 157 in cattle, which go a long way to eliminating some of the environmental concerns that we have, and cross-contamination of not only milk but fresh fruits and vegetables, which is a problem in the US and Canada So that's a little bit about our probiotic bioactives. Perhaps lastly, I'm going to talk a little bit about, it was mentioned this morning about bacteriophage. Bacteriophage are viruses that are specific to bacteria, and they work by, they again have a syringe-like system. They inject their DNA into bacterial cells or RNA into bacterial cells. They make multiple copies of themselves. They take over the machinery of the bacterial cells to produce several hundred copies of themselves. They then lyse the host cells, and they are released to go out and find some more bacteria to kill those. So they have an innate specificity, so you can get bacteriophage that will only infect Salmonella, will only infect, for instance, E. coli 157, and there's a range of bacterial viruses against most of the common bacterial foodborne pathogens. They have GRAS status that's generally recognized as safe status with FDA and USDA, and they are also approved for use by EFSA and by Health Canada. And they actually have been certified for use in organic production systems. What we've been looking at is, instead of using these as a sort of a spray, which is the usual way of handling bacteriophage application, we've been looking at possibly using bacteriophage in packaging material. Just some work that we did a fair number of years ago now. We had a fairly major Salmonella Enteritidis outbreak in Canada, which was associated with cheddar cheese. What we were interested to do to see if we could use bacteriophage to control Salmonella, both in cheeses made from raw milk and pasteurized milk. So the top two lines show Salmonella survival in the cheese without the application of phage, and the bottom two show survival in the presence of phage. And there's some indication that phage work a little bit better in pasteurized milk cheeses than in normal cheeses. But there's certainly about a 2-log reduction in levels of Salmonella initially in the milk, and it does affect survival in certain cheeses. There are a couple of companies that are producing phages that are active against Listeria monocytogenes. This is work that was done with the Listex phage, not in our lab, but again shows that Listeria monocytogenes can be used to control, Listeria monocytogenes phage can be used to control the growth of Listeria monocytogenes in soft cheeses. So this is days after cheese making. These are the controls, and this is with phage addition. So again, there's good evidence to suggest that bacteriophage can be used to control pathogens in cheeses. Again, it's maybe a sort of a strange concept for cheesemakers to actually think about using bacteriophage, because generally bacteriophage, you don't want bacteriophage in your plants, but there are some good bacteriophage as well. So what we've been doing is taking bacteriophage and developing ways of sticking them to membranes, paper-based membranes. So the idea is that we immobilize them in a very unique way. So that we immobilize them head down. So this is the tail region. So this is the region that sticks to the surface of the bacterial cell. So we want that to remain active. And what happens is that These phage then capture the target cell. They remain infective, so they can then inject their nucleic acid, propagate, and kill the bacterial cell. And this is some photos showing some bacteriophage that are immobilized on surfaces. So you can see that, can't see the tails very well, but you can see that the heads are sticking to the surface of the material. And we haven't really done much work with cheeses yet, but we've shown that in ready-to-eat meats, we can control listeria in aerobic environments, in modified atmosphere packaged environments, and in vacuum packaged environments at 4 degrees centigrade. So we've got some good evidence that we can, the bottom, this is our phage membranes, our phage packaging material. So we've got a control of the growth of listeria in ready-to-eat meats. And again, this is a visualization of, again, using our immobilized phage, our phage packaging material, to control E. coli 157H7 on raw meat. Again, we're using our luminescent E. coli. So this is the packaging material without the phage, and this is the phage containing membrane. So you can see that, again, the colored areas represent growth of E. coli 157. With our packaging material, with phages, we don't get any growth of E. coli 157. So again, a link to the use of microorganisms to improve safety of products. So it's not just me. I'm not going to talk about the non-thermal processing work that we're doing. I think I've run out of time. So, but it's not just me. I just get to come to nice places like North Canterbury and talk about this. All these cool guys do all the work, and I get all the credit. So it's been a whole bunch of gifted researchers that have been involved in this work. So I'll leave it at that.